Welcome to this presentation on organisms respond to changes in their internal and external environments. Organisms must detect and respond to changes in both their internal and external environments. These responses help maintain survival, reproduction, and stable conditions within the body. Stimuli, responses, and survival value, receptors and sensory detection. A stimulus is defined as a detectable change in the internal or external environment of an organism that produces a response. External stimuli include factors such as light intensity, temperature, pressure, and the presence of chemicals like nutrients or toxins. Internal stimuli arise from within the organism, for example changes in blood glucose concentration, carbon dioxide levels in the blood, or body temperature. A response is any change in behavior or physiology that occurs as a result of a stimulus, and these responses are essential for survival because they allow organisms to maintain optimal conditions for enzyme-controlled reactions. Without effective responses, cells would be exposed to harmful conditions that reduce metabolic efficiency or cause damage. Responses have clear survival value and are shaped by natural selection. For example, a rapid withdrawal of the hand from a hot surface reduces tissue damage and increases the chance of survival. Similarly, mammals responding to cold conditions by shivering generate heat through increased respiration in muscle cells, helping maintain a stable core temperature. In plants, responses such as phototropism, where shoots grow towards light, increase light absorption and therefore the rate of photosynthesis, supporting growth and reproduction. These examples show that responses are not random, they are adaptive and improve fitness. Receptors are specialized cells or groups of cells that detect stimuli and convert them into electrical signals. Receptors function by transducing energy from a stimulus into a generator potential, which may then lead to an action potential. Different receptors are adapted to detect specific types of stimuli, ensuring sensitivity and accuracy. For example, photoreceptors in the retina detect light, chemoreceptors detect chemicals, mechanoreceptors respond to pressure or movement, and thermoreceptors detect temperature changes. Photoreceptors, such as rod and cone cells in the human retina, contain light-sensitive pigments that change shape when they absorb photons. This triggers a sequence of events leading to a change in membrane potential. Rod cells are highly sensitive and function well in low light, while cone cells require higher light intensities and provide color vision. Chemoreceptors play a crucial role in detecting substances such as oxygen, carbon dioxide, and pH. In humans, chemoreceptors in the carotid bodies monitor blood chemistry and help regulate breathing rate. Mechanoreceptors respond to physical deformation of the receptor cell membrane. Examples include Pacinian corpuscles in the skin, which detect pressure and vibration, and hair cells in the cochlea, which are essential for hearing. Thermoreceptors, found in the skin and hypothalamus, detect changes in temperature and contribute to thermoregulation. Each receptor type has a specific threshold. Only stimuli above this threshold generate a response, preventing constant activation by insignificant changes. The nervous system and response pathways, structure and function of neurons and synapses. The nervous system enables rapid communication and coordination in animals, allowing them to respond quickly to stimuli. In mammals, the central nervous system, CNS, consisting of the brain and spinal cord, acts as the coordinator. It processes information received from receptors and determines the appropriate response. The peripheral nervous system connects receptors and effectors to the CNS via sensory and motor neurons. Sensory neurons transmit electrical impulses from receptors to the CNS, while motor neurons carry impulses from the CNS to effectors such as muscles or glands. Relay neurons, located within the CNS, link sensory and motor neurons, allowing integration and processing of information. This organization ensures that responses are coordinated rather than isolated, enabling complex behaviors such as movement, communication, and learning. Reflexes are rapid, automatic responses to stimuli that do not involve conscious thought. They are protective, reducing the risk of injury. The reflex arc includes a receptor, sensory neuron, relay neuron, motor neuron, and effector. For example, in the withdrawal reflex, pain receptors in the skin detect a harmful stimulus, and the impulse travels via the spinal cord to motor neurons that stimulate muscle contraction, pulling the hand away. Neurons are specialized cells adapted for the rapid transmission of electrical impulses. Sensory neurons carry impulses from receptors to the CNS and have a long dendron and short axon. Motor neurons transmit impulses from the CNS to effectors and typically have a long axon. Relay neurons are found entirely within the CNS and connect sensory and motor neurons. The structure of neurons supports their function. The axon is often myelinated by Schwann cells, forming a myelin sheath that acts as an electrical insulator. This allows saltatory conduction, where impulses jump between nodes of Ranvier, significantly increasing transmission speed. The cell surface membrane contains sodium and potassium ion channels essential for generating action potentials through changes in membrane permeability. Synapses are junctions between neurons that enable communication. Transmission across a synapse is chemical, involving neurotransmitters such as acetylcholine. When an action potential reaches the presynaptic terminal, calcium ion channels open, causing vesicles containing neurotransmitter to fuse with the presynaptic membrane and release their contents into the synaptic cleft. The neurotransmitter diffuses across and binds to receptors on the postsynaptic membrane, leading to depolarization if excitatory. Synapses ensure one-way transmission because neurotransmitter is released only from the presynaptic neuron, and receptors are present only on the postsynaptic membrane. They also allow summation, where multiple impulses can combine to trigger a response, and inhibition, preventing overstimulation. Skeletal muscles as effectors. Effectors are cells or tissues that carry out responses, and in animals these include muscles and glands. Skeletal muscles are particularly important effectors, enabling movement in response to nervous stimulation. Muscle fibers are long, multinucleate cells containing myofibrils. These myofibrils are made up of repeating units called sarcomeres, which contain thick myosin filaments and thin actin filaments. Muscle contraction occurs when these filaments slide past each other, shortening the sarcomere and therefore the muscle. This process does not involve the filaments changing length, but rather overlapping to a greater extent. The sliding filament mechanism is triggered by nervous stimulation. An action potential arrives at the neuromuscular junction, causing the release of acetylcholine. This depolarizes the muscle cell membrane and leads to the release of calcium ions from the sarcoplasmic reticulum. Calcium ions bind to troponin, causing tropomyosin to move and expose myosin binding sites on actin. Myosin heads form crossbridges with actin and, using energy from ATP, pull the actin filaments towards the center of the sarcomere. ATP is essential for muscle contraction, providing energy for crossbridge formation and detachment. Without ATP, muscles would remain rigid, as seen in rigor mortis. Principles of homeostasis and negative feedback, control of blood glucose concentration. Homeostasis is the maintenance of a stable internal environment within narrow limits, despite changes in the external environment. Variables commonly regulated include body temperature, blood glucose concentration, blood pH, and water potential. Homeostatic systems operate through negative feedback mechanisms. In negative feedback, any deviation from the set point is detected by receptors, and corrective responses are initiated to reverse the change. This prevents conditions from becoming too extreme. For example, if body temperature rises above normal, mechanisms such as sweating and vasodilation increase heat loss, returning temperature towards the set point. A typical negative feedback loop involves receptors, a coordination center, and effectors. The coordination center, such as the hypothalamus in thermoregulation, compares incoming information with the set point and sends signals to effectors. Once the variable returns to normal, the corrective response is reduced or stopped. This self-regulating system ensures stability without constant external input. Negative feedback contrasts with positive feedback, where change is amplified rather than reversed. Positive feedback is rare in biological systems and usually associated with specific events, such as childbirth. Blood glucose must be maintained within narrow limits to ensure a constant supply of energy for respiration, particularly for the brain, which relies almost exclusively on glucose. This regulation is achieved through the antagonistic actions of the hormones insulin and glucagon, produced by the pancreas. When blood glucose concentration rises, such as after a carbohydrate-rich meal, beta cells in the islets of Langerhans detect this change and release insulin. Insulin binds to receptors on target cells, especially liver and muscle cells, increasing the uptake of glucose. It also stimulates the conversion of glucose to glycogen in the liver and muscles, a process known as glycogenesis. As a result, blood glucose concentration falls back towards the normal level. Conversely, when blood glucose concentration falls, alpha cells in the pancreas release glucagon. Glucagon stimulates the breakdown of glycogen to glucose in the liver, known as glycogenolysis, and the production of glucose from non-carbohydrate sources, or gluconeogenesis. This raises blood glucose concentration back to the set point. This system operates through negative feedback, with insulin and glucagon acting antagonistically. Failure of this system can lead to diabetes mellitus. Tropisms and plant growth responses. Plants respond to environmental stimuli through growth responses known as tropisms. Phototropism and gravitropism are directional growth responses to light and gravity respectively. These responses are slower than animal responses but are essential for maximizing photosynthesis and water uptake. Phototropism in shoots is usually positive, meaning shoots grow towards light. This response is controlled by the plant growth regulator auxin, produced in the tips of shoots. Auxin is redistributed to the shaded side of the shoot, where it stimulates cell elongation. As cells on the shaded side elongate more than those on the illuminated side, the shoot bends towards the light. This increases light absorption and enhances photosynthesis. Gravitropism involves growth in response to gravity. Roots show positive gravitropism, growing towards gravity, while shoots show negative gravitropism. In roots, auxin accumulates on the lower side due to gravity. However, in roots, high concentrations of auxin inhibit cell elongation, causing the upper side to grow faster and the root to bend downwards. This ensures roots grow into the soil, improving anchorage and water absorption. These mechanisms demonstrate that plants are highly responsive organisms, despite lacking a nervous system. Responses rely on chemical coordination and differential growth. Organisms rely on receptors, coordination systems, and effectors to respond to environmental changes. These responses maintain homeostasis and increase chances of survival. Thank you for watching.